What I wish I knew at the start of my gardening journey. Plant tissue culture, ready to unlock its secrets. Plant tissue culture is like plant cloning on steroids. It's a method where tiny pieces of a plant called tissues are grown in a nutrient-rich medium. Imagine creating a mini plant factory in a jar. First, you take a small section from a plant. This could be a leaf, stem, or even root. Then, you place it in a special gel filled with nutrients and hormones. Under the right conditions, this tissue grows into a whole new plant. Why is this cool? Because you can grow lots of plants quickly, all genetically identical to the original, perfect for rare plants or those tricky to propagate. So, next time you see a beautiful plant and wish you had more, remember, with plant tissue culture.
Want to cultivate plants from tiny tissues? Here's how to prep the perfect growth media. First, gather your materials: distilled water, agar, plant nutrients, and a pH meter. Start by measuring the right amounts of nutrients based on your plant's needs. Mix them with distilled water in a beaker, stirring until fully dissolved. Next, add a pinch of agar to help solidify the mixture. Heat the solution gently, stirring until the agar completely melts. Now, use your pH meter to check the solution's pH level. Adjust it to between 5.6 and 5.8 using a few drops of acid or base. Pour the mixture into sterilized containers and let it cool until it solidifies. And voila! You've just prepared your own growth media for plant tissue.
You need to hear this if you're working in plant science. The key to thriving tissue cultures might be simpler than you think. Water quality is the game changer in tissue culture. It's the largest component of your media, so getting it right is crucial. Let's break down the top 3 purification methods. First up, distillation. This process boils water, then condenses the steam, leaving impurities behind. It's super pure but energy intensive. Next, deionization. This method uses resin beads to swap out unwanted ions. It's efficient, but regular maintenance is a must. Then there's reverse osmosis. This technique pushes water through a membrane to filter out contaminants. It's effective and widely used, but wait, there's more. Rainwater can be a fantastic alternative. Collect it from a non-metal roof into a plastic container. Ensure your total dissolved solids, TDS, are between 0-10 and pH is 6.8-7. Remember, the right water makes all the difference. Cheers to perfect tissue.
Exposing the secret to flawless measurements in labs. It's simpler than you think. When it comes to laboratory work, accuracy is non-negotiable. Enter the weighing balance, your indispensable ally in precision. From 0.0001 grams to 210 grams, these balances handle it all. They're not just about numbers. Need to prepare a sample accurately? Check. Measuring ingredients for that pharmaceutical breakthrough? Double check. But wait, there's more. Weighing balances are pivotal in measuring food and beverage products, ensuring quality in manufacturing, and even diving deep into biological studies. In the realm of material engineering and pollution level analysis, precision can make or break results. That's why every lab, from the smallest research nook to the grandest facility, needs a weighing balance.
What's the secret weapon for every chemist? Spoiler alert, it's the magnetic stirrer. Alright, so what exactly is a magnetic stirrer? Picture this, a small device that uses a rotating magnetic field to spin a stir bar inside your liquid. Simple, right? But super effective. This nifty gadget is a game changer in the lab. Why? Because it makes stirring small volumes a breeze. No more manual stirring with rods or dealing with bulky overhead stirrers. Magnetic stirrers are perfect for surface sterilization and preparing growth media. They ensure your solution is consistently mixed, giving you uniform results every time. And if your mixture needs heating, no worries. Hot plate magnetic stirrers are here to save the day, combining heating and stirring in one sleek package. So chemists, next time you're in the lab, give a nod to the unsung hero on your bench. The magnetic stirrer, small but mighty, and an absolute...
This is gonna blow your mind. Your tissue culture's success might just hinge on something as simple as pH. pH stands for the potential of hydrogen. Now, it's a scale from 1 to 14 that tells us how acidic or alkaline a solution is. Acidic solutions range from 1 to 5.5, neutral solutions are between 5.6 and 7, and alkaline solutions span from 7.1 to 14. Now, why does this matter in tissue culture? Different plants thrive at different pH levels. For example, Rhododendrons prefer a more acidic environment with a pH of 4.5 to 5. On the other hand, strawberries do better in a slightly less acidic setting, around pH 5.7. Most tissue cultures are grown in media with a pH of a 5.2 to 5.8. Before you start, you need to adjust the pH, especially before autoclaving. Whether you're using agar or not, getting the pH right is crucial for plant growth. So next time you're prepping your culture media, remember, pH isn't just a number. It's the key to healthy, thriving.
Ever thought a pressure cooker could be your plant's best friend? Well, get ready to be amazed. In plant tissue culture, keeping everything sterile is a game changer. But did you know you can use a pressure cooker as an autoclave? Yup, that trusty kitchen gadget can help you kill those pesky microbes. Here's how it works. A pressure cooker superheats water by increasing steam pressure, just like an autoclave. The magic number? 15 psi. At this pressure, water boils at 121 degrees Celsius, or 250 degrees Fahrenheit. Perfect for sterilizing your growth media and equipment, like surgical knife, tweezer, petri dishes, beaker, and even tissue paper. First, add an inch or two of water to the pressure cooker. Once it hits its highest pressure, start your timer for 25 minutes.
If your dream is to grow plants like a pro, you're in luck. Plant tissue culture has been a booming field of science over the last few decades. Nowadays, its knowledge is so readily available that anyone with genuine interest can do it. In the long run, it can generate enormous financial returns. Let's set up your very own tissue culture lab. First things first, location matters. Choose a spot away from air vents, bacteria, and insect labs to avoid contamination. Now, let's break down the essentials. 1. Quarantine area. Keep it clean. Change your shoes and lab coat here to prevent bringing in contaminants. 2. Washing area. Wash and dry your plant materials and lab gear. You can clean your used lab gears also for reuse. 3. Media preparation area. Mix your culture media with tools like magnetic stirrers, pH meters, and autoclaves. 4. Transfer area. This sterile zone is where the magic happens. Install laminar flow cabinets to keep air filtered and clean, most preferably HEPA filter. 5. Culture grow room. Maintain perfect conditions for your plants with controlled temperature, humidity, and avoid direct sunlight. So this room must be out of any window or vent. And finally, data collection area. Keep track of your experiments with a reliable computer setup. With these areas in place, your tissue culture lab will be up and running smoothly. Ready to grow your business and your plants. Let's get started.